everyone. Today I'm excited to delve into the fascinating research study that explores the unique mechanisms of gene expression in Trypanosoma Cruzi, which is a parasite known to cause Chagas disease. The focus of our discussion will be on the intriguing role of translation control in this particular process. The research study we're discussing today is titled Ribosome Profiling Reveals Translation Control as a Key Mechanism, Generating Differential Gene Expression in Trypanosoma Cruzi. This study was conducted by a team of researchers and published in the BMC Genomics Journal in 2015. The researchers used a technique known as ribosome profiling to study the translation process in Trypanosoma Cruzi, and their findings provided a new insight into the unique mechanisms of gene expression in this parasite. But before we dive in, let's take a look at what we'll be covering today. We'll start with some background and context, followed by an overview of what Chagas disease is and why it's important to research it. Then we'll introduce the specific research study we'll be discussing today. After that, we'll dive into the methodology of this study, discussing its findings, explore the implications of these results. At the end, we'll wrap up with a conclusion, and I'll be taking any questions via our Journal Club discussion board at the very end. Let's start with some background information to set the stage for our discussion. In the world of biology, the process of gene expression, which is how information from a gene is used to create a functional product like protein, is typically controlled at a stage known as transcription. This is where DNA, as you know, the blueprint of life, is converted into RNA. However, in the case of Trypanosoma cruzi, this transcription process is not regulated, which is quite unusual. This led the researchers to hypothesize that T.cruzi must be controlling gene expression at a later stage, specifically at the translation stage, where RNA is converted into protein. This unique mechanism of gene expression control is the focus of our discussion today. Moving on to the next slide, we'll talk about Chagas disease, which is caused by the Trypanosoma cruzi parasite. This disease is transmitted to humans by insects known as kissing bugs. The symptoms of Chagas disease can vary widely, ranging from mild and short-term to severe and chronic. If left untreated, it can lead to serious heart and digestive problems. However, despite its significant impact on public health, Chagas disease is still considered a neglected tropical disease, meaning it has historically received less attention and research funding compared to other diseases. However, studying T.cruzi and its unique mechanisms of gene expression can enhance our understanding of this parasite and potentially lead to the development of more effective treatments for Chagas disease. As we move to the next slide, we see a map showing the areas more affected of Chagas disease. The countries highlighted here in red are the most vulnerable to this disease, as you can see. Chagas disease is a major health concern, especially in Northern and especially in Latin America. This geographical distribution underscores the importance of our research in T.cruzi by understanding the biology of this parasite and how it controls gene expression. Then we can contribute to the global effort to combat Chagas disease and reduce its impact on these vulnerable populations. Now, let's return our attention to the specific study we'll be discussing today. The study, again, is titled Ribosome Profiling Reveals Translation Control as the Key Mechanism Generating Differential Gene Expression in Trypanosoma Cruzi. This research was published again in the BMC Genomics Journal in 2015. The researchers in the study use a technique known as ribosome profiling, which we'll talk about later. This technique allows us to see which parts of the RNA are being read by the ribosome to produce proteins, providing a snapshot of the translation process in action. The study compared two life stages of the parasite, the non-infective stage, known as the epimastigote stage, and the infective stage, known as the metacyclic tryptomastigote stage, or also referred to as MT for short. the goal was to understand how T.cruzi controls gene expression at the translation stage and how this control varies between different life stages. This research is significant because it challenges our traditional understanding of gene expression control and provides new insights into the biology of T.cruzi. So, as we move through this presentation, we'll dive deeper into the methodology of the study, discuss its key findings, and explore the implications of these results. So, now for the methodology of the study. This is the how of the research, the techniques, and the processes the researchers used to gather and analyze their data. Understanding the methodology is crucial because it not only provides context for the findings, but also allows us to assess the validity and reliability of the results. In this section, we'll explore the main techniques used in the study and discuss how the data was analyzed. First, let's take a look at the overall research phases. The researchers, they started with a thorough review of existing literature on Trypanosoma Cruzi and its gene expression, which led them to formulate their hypothesis. They then controlled data using RNA sequencing and ribosome profiling and analyzed this data to identify differences in gene expression and translation between the two life stages of the parasite. Their interpretation of the results led to the conclusion that translation regulation plays a major role in the parasite's differentiation. The findings were then finally published in the journal BMT Genomics in 2015. As we dive deeper into the methodology of the study, let's first focus on the main technique used, ribosome profiling. This technique is used to measure the translation of mRNA into proteins, also known as the translatome. This technique involves isolating the ribosomes from the cells. Ribosomes, as you may know, are the molecular machines in our cells that can read RNA and build proteins. By sequencing the RNA fragments that are being read by the ribosomes, the researchers were able to take a snapshot of the translation process in action. In the study, ribosome profiling was used to determine how much of the mRNA was being translated into protein in both stages of the parasite. In parallel with ribosome profiling, the researchers also performed mRNA sequencing. This technique is used to measure the levels of mRNA in the cells, also known as a transcriptome. In context of the study, RNA sequencing was used to determine the levels of mRNA in both the non-infective and infective life stages of Trypanosoma Cruzi. Now, by comparing the total mRNA with the mRNA that is being actively translated into the protein from ribosome profiling, or like we said, translatome, the researchers could identify which genes are being regulated at the translation stage. So basically, to summarize, RNA sequencing measures the transcriptome, which is mRNA levels, and ribosome profiling measures the translatome, which is the translation of mRNA into proteins. The researchers used these two powerful techniques to get a comprehensive view of gene expression in Trypanosoma Cruzi. This approach allowed them to uncover the unique role of translation in this organism. And now comes the data analysis. This is where the researchers took the raw data from the ribosome profiling and mRNA sequencing and began to decode it. First, they performed bioinformatics analysis on the data, which involved identifying which genes were being translated and at what levels. They then compared this data between the non-infective and infective stages of the parasite, which is our control variable in this research. This allowed them to analyze how the translation process changes as a parasite prepares to infect a host. As for the specific methods used in this data analysis, their procedure began with pre-processing, which involved quality control and trimming of the sequencing reads. This ensured that the data was clean and ready for analysis. Next, they performed mapping and alignment. This involved matching the sequencing reads to the genomic information of T.cruzi, and they used a tool called CLC Genomics Workbench 6.5. Once the reads were mapped and aligned, the researchers quantified gene expression. This involved counting the reads or estimating the levels of genetic material. They then analyzed translation efficiency by comparing mRNA and translatome levels. This gave them insights into how efficiently the genes are being translated into proteins. Then statistical analysis was performed to identify significant differences in expression or translation. This involved using statistical tests to determine whether the observed differences were due to chance or represented real differences. And finally, the researchers visualized and interpreted the data. They created visual representations like heat maps, scatter plots, box plots to better understand the data. So, as you can see, the data analysis was a complex and multi-step process, but it was crucial in uncovering the key findings of the studies. And speaking of findings, that brings us to the next section of this presentation. Okay, so having explored the methodology of the study, now we turn to the heart of the matter, the findings. This is where we decode the data and unveil the results of the research. The findings section is where we'll answer the question, what did the researchers discover about gene expression and translation control in Trypanosoma cruzi? In this section, we'll delve into the details of the study's results, from mRNA levels and the translation process, to the correlation with the proteome and the efficiency of translation. In this slide, we're looking at the mRNA levels in the two main stages of the T.cruzi life cycle. While the overall levels of mRNA are similar in both stages, the study found that about 26% of the transcripts detected showed at least a twofold change in gene expression levels between the two stages. This suggests that while transcription is constitutive, meaning it occurs continuously, there are significant differences in the expression of specific genes. These differences are likely due to post-transcriptional regulation, which is a process that controls gene expression after the transcription stage. And this is illustrated in a figure 1, where we can see a Venn diagram showing similarity in the number of detected genes in each stage. A scatter plot displayed the estimated mRNA expression levels for both stages, and a heat map illustrating the variation in expression for the genes, showing differences at the mRNA level in each stage. In the exploration of translation process in Trypanosoma cruzi, which is one of the key findings that is a significant portion of the translatome, which is the entirety of the mRNA molecules being translated, is downregulated in the infective stage, or MT. This means that fewer proteins are being produced from the mRNA in this stage. This downregulation suggests that the control of the gene expression in T.cruzi occurs at the translation stage and not the transcription stage. And this is a significant finding because it challenges our previous understanding of gene expression of this organism. And furthermore, the researchers believe that this downregulation of translation could be a strategic move by the parasite. By reducing the production of proteins, the parasite might be preparing itself for the infective stage, or MT, conserving resources and energy. If we take a look at figures 2 and 3, we can see a visual representation of these findings. Figure 2 shows the number of detected genes in each stage, with the differentially expressed genes highlighted. Figure 3, on the other hand, displays the estimated translation levels for both stages, providing a clear picture of the downregulation that occurs in the infective stage, again, the MT stage. In conclusion, examination of the translation process in T.cruzi has revealed that translation regulation plays a crucial role in the life cycle of this parasite, particularly in its transition to the infective stage. Now, let's move on to the correlation between protein levels and translation levels. This is where we'll connect the dots between the translitome and the proteome. The researchers found that the level of proteins in the parasite correlated better with the levels of translation, also known as the translitome, than with the levels of transcription, or the transcriptome. This supports the idea that gene expression control in T.cruzi occurs at the translation stage. Figure 4 in this research article provides a visual representation of this correlation. The scatter plots in this figure show the correlation of the proteome to the transcriptome and the translitome for the E and the MT stages. It can be clearly observed that translitome data are better correlated to protein expression than transcriptome levels. This is especially true for the epimastogote stage, where the correlation coefficient between translitome and proteome goes up from a correlation coefficient of 0.41 for the transcriptome to be 0.80 for the translitome. This correlation is a significant piece because it indicates that the level of translation rather than the level of transcription is a better predictor of protein levels in T.cruzi. This further emphasizes the importance of translational regulation in the life cycle of this parasite. And now we'll talk about translation efficiency. Figure 5 from the research article provides a comprehensive view of this concept. Translation efficiency is defined as the number of ribosome footprints per transcript, giving an idea of ribosome occupancy per messenger molecule. The researchers observed large differences in translation efficiency among transcripts in the same life cycle stage between the transcript and the two life cycle stages. This suggests that the regulation of translation efficiency constitutes a means to rapidly adjust the yield of specific protein products from the available mRNA steady-state levels. Using a twofold change as a determination of differential expression, they found that 643 genes are regulated exclusively at the level of translation upon differentiation. These results further support the importance of translation in the regulation of stage-specific gene expression. And overall, these findings provide valuable insights into the regulation of gene expression in T.cruzi and highlight the importance of considering translational efficiency for a more accurate understanding of protein expression levels. The regulation of translation efficiency may play a crucial role in the parasite's adaptation and response to its environment, particularly during the transition from the vector's gut to the mammalian host during the infective MT stage. And now we'll talk about what specific gene families are regulated at the translation level in Trypanosoma Cruzi. The regulation of these gene families is crucial for the parasite's adaptation and survival in the different stages of its life cycle. Figure 6 in the research article provides a visual representation of this regulation. It shows that translation efficiency fold change, or MT over E, for different gene families in the MT and E stages. Let's focus on the transilidase superfamily, or TS for short. In the MT stage, gene coding for members of the TS superfamily are the most overrepresented among the ones with a translation efficiency fold change, or MT over E, higher than 2. This family of virulence factors plays a significant role in the infectivity of T.cruzi. The TS family members are responsible for transferring sialyl residues from the host, and a high number of the family members are expressed in the MT stage. By coating itself with the host's own molecules, the parasite can avoid detection and destruction by the host's immune system. This is one of the reasons why TS superfamily is considered a key virulence factor in T.cruzi. Interestingly, the translation efficiency of the TS family in the MT stage is significantly higher than in the E stage. This increase in translation efficiency among differentiation to the MT stage is in accordance with previous reports, which further emphasize the importance of translational regulation in T.cruzi. On the other hand, in the MT stage, the genes coding for ribosomal proteins, or RP, are the most overrepresented among the ones with the translation efficiency fold change. Again, MT over E, which is lower than 0.5. This downregulation is mainly derived from the low translation levels of the RP genes in the MT stage, suggesting a possible reduction of ribosomal RNA synthesis consistent with a downregulation of ribosomal protein production. So, as we reach the final section of our presentation, it's time to reflect on the key findings from this study on gene expression in Trypanosoma Cruzi. We've covered a lot of ground today, so let's take a moment to summarize the key findings from this research article on gene expression in Trypanosoma Cruzi. Firstly, we learned that the levels of mRNA were similar in both the non-infective and infective stages of the parasite. This suggests that the control of gene expression does not occur at the transcription stage, but rather post-transcriptional regulation is responsible for the majority of gene expression changes in trypanosamids. We then delved into the translation process, where we found that translation was downregulated in many genes in the infective stage. This indicates that control of gene expression does occur at the translation stage, as they hypothesized. Next, we explored the correlation between protein levels and transcription levels, finding that the levels of protein in the parasite correlated better with the levels of translation, also known as a translatome, than with the levels of transcription, or the transcriptome. We also discovered that the translation efficiency is highly variable for different mRNAs in both life stages of the parasite, and a significant number of genes change their translation efficiency upon differentiation. And finally, we examined specific gene families, such as the TS family, which had a higher translation efficiency in the infective stage, which could help the parasite to infect their host cells. In conclusion, the study provides valuable insights into the regulation of gene expression in T.cruzi and highlights the importance of considering translational efficiency for a more accurate understanding of protein expression levels. The regulation of translation efficiency may play a crucial role in the parasite's adaptation and response to its environment, particularly during its transition from the vector's gut to the mammalian host during the infective MT stage. As we wrap up our discussion, it's important to consider the limitations of this study and the directions for future research. Firstly, there are technological limitations. The methods used in the study, such as RNA sequencing and ribosome profiling, while advanced, are not perfect and may not capture all of the nuances of gene expression and translation. For example, they may not fully account for post-translational modifications or the activity of non-coding RNAs. Secondly, the interpretation of the results relies on the assumption that mRNA levels and their translation are the main factors controlling gene expression in Trypanosoma Cruzi. However, other factors such as protein degradation, post-translational modifications, or epigenetic changes could also play significant roles. Therefore, the results should be interpreted with caution. In terms of future research, the findings for the study upon several interesting avenues. For instance, further studies could delve into the specific mechanisms of post-transcriptional regulation in T.cruzi. Additionally, research could also focus on the role of specific gene families, such as the trans-sialidase family, and the infectivity of the parasite. Understanding these mechanisms could potentially lead to the development of new treatments for Chagas disease. And lastly, I want to leave you with a question to ponder. Given the importance of translational regulation in T.cruzi, how might this knowledge be applied to other parasites, or even to our understanding of gene expression in other organisms. What's your take? I hope this presentation has provided you with a deeper understanding of the regulation of gene expression in Trypanosoma cruzi and its implications for the parasite's life cycle and infectivity. I'm looking forward to hear your thoughts and answering any questions you may have. Thanks.